Hello, this is Carsten Bönnemann from the NIH, and I want to give you a little introduction into what precision therapeutics mean for the collagen 6 disorder and introduce the concept of a gene therapy toolbox to address these disorders. We talk about gene-directed approaches to congenital disorders of muscle, in particular disorders of collagen 6. It is driven by a paradigm and a promise, which is the closer we get to the origin of the disease, the higher we expect the impact to be. Let me explain that in this little diagram here. That's very simply showing that the genes are written in DNA on the chromosome. They're then read by the transcription machinery that put the bits and pieces of the genes that are called exons together in a contiguous message called the messenger RNA, which is then translated and made into protein, which then leads to a person. In a genetic disease, we have the fault directly in the DNA, in the way the genes are encoded in DNA, and that's a mutation or a pathogenic variant in DNA, and that's carried forward because it's transcribed into the messenger RNA, resulting in a faulty or even absent protein, and that results in disease. The promise of gene-directed therapies is that the closer we go to the actual origin of the disease in the DNA, the better the impact is expected to be. So if we're up here in the DNA or the transcript where the mutation is occurring before it's made into protein or even into a patient, the higher the impact is could be. That doesn't negate that medical care is super important all along in treating these disorders, and I will show you how important that is, in particular in the collagen 6 disorders. What precision medicine really means is that we want to directly address precisely the gene where the condition originates, and sometimes even the precise mutation that occurs in an individual patient. So for the precision therapeutics to be developed, we need precision diagnostics first. So we need to be precise about the diagnosis in order, down to the genetic level, in order to be precise about the treatment. This is an example of a muscle biopsy in a child with collagen 6 congenital muscular dystrophy. You can see the features of the muscular dystrophy, or the degeneration and regeneration that happens over time progressively, with muscle fibers being encased now in connective tissue. You can see the cell nuclei here in the center of the fiber, meaning regeneration, and loss of fibers all around. And so it's progressive over time, and we need to be aware that we have to intervene as early as we can. The congenital muscular dystrophies, interestingly, are disorders of the muscle extracellular matrix, meaning it's the covering of the muscles, and that gives anchorage of the muscle into the extracellular space that is involved in the congenital muscular dystrophies. Here's a membrane of a muscle cell. Here's a protein complex that links the muscle from the inside to the outside. And these three groups are the most important congenital muscular dystrophy: dystroglycanopathy, laminin 2-1-1 and then, of course, collagen type 6, which is really right here in the extracellular matrix connecting to the surface of the muscle cell from the outside. There are 3 genes to be reckoned with: collagen 6 alpha 1, 2, and 3, and we can have 2 different genetic mechanisms, dominant or recessive, and that is to be taken into account when we think about therapeutics. The way collagen 6 is put together is fairly complex, but important to understand. So I told you collagen 6 genes are on the chromosomes, and then it's made into a message, and the message is being made into a protein, which is, you can see here, and we have the alpha 1, the alpha 2, and the alpha 3 chains that are now proteins, and they are being assembled into a bigger protein that now contains all 3 chains. We call that monomer, heterotrimeric monomer, it's made into a dimer, it's made into a tetramer, and the tetramer now contains 4 of these monomers with 3 chains each, so 12 different polypeptides, and those are secreted into the matrix. Interestingly, not by the muscle cells, but by connective tissue cells living in between the muscle cells. We call them interstitial fibroblastic cells, or in modern terminology, FABs, or fibroadipogenic progenitors, FABs here. And we get back to them, they're really important. This is what collagen 6 looks like under the electron microscope. You can see how these tetramers now are lined up very nicely like beads on a string in the matrix. Collagen 6 disorders, as you well know, are a spectrum of conditions from the more severe congenital muscular dystrophy type Ullrich, described by Otto Ullrich in the late 20s and early 30s in Germany, but then also milder with intermediate phenotypes, and then also a Bethlem-type muscular dystrophy that is considerably milder. You see the typical features that you all know in children born with this condition, where you have joint contractures as well as joint hyperlaxity coexisting, as well as muscle weakness, often scoliosis, and then difficulties with ambulation as the disease progresses. I told you medical care is always all important, and it is really also a form of precision therapy, because medical care in the congenital muscular dystrophies, particularly in type Ullrich, is really driven and informed by our knowledge about disease mechanism and natural history. Here you see a child breathing in an MRI scanner, and we can see the diaphragm moving up and down, but it's not moving normally. It's actually quite weak in its movements. So this muscle here, hidden in the chest, the diaphragm, is quite weak early on, and that causes early respiratory difficulties, respiratory insufficiency, that you can see here in Regan Foley's natural history plotted over the first 10, 20 years of life. You can see how the forced vital capacity, a measure of lung function, declines very predictably over the years. And by the late teens, early 20s, everyone is in need of respiratory support. When respiratory support is provided, though, then the natural history of this disease dramatically changes, and life expectancy changes from no longer than the 20s to much, much longer. We don't even know where the limits are. There are 2 major genetic mechanisms that underlie collagen 6 related dystrophies, and they have to be treated separately when we talk about therapeutics. There's a loss of function mechanism where we have mutations on both copies of the gene. There's one copy from dad, one copy from mom. And when both copies carry mutations, then there is no normal collagen 6 that can be made, and that's often a loss of collagen 6 production altogether. But then there's also a different mechanism which actually is more common, which is dominant negative, where the mutation is only on one of the copies, but it now is toxic. It interferes with the normal collagen 6 from the normal copy of the gene, and then causes havoc that way. Under the microscope, it looks like this. In the recessive cases, collagen 6 is red here. In the recessive cases, no collagen 6 there because it's loss of functions. No collagen 6 is being made. If you have one abnormal copy that's a loss of function copy, so a carrier for a recessive mutation, but have a normal copy, you are actually unaffected. You're clinically normal. This is a gentleman here who carries loss of function mutation on 2 of the collagen 6 genes called 6A1 and A2 and still is unaffected. And the more common mechanism of dominant mutations looks like this. Collagen 6 is out there in red here in the muscle matrix, but it doesn't connect to the muscle fibers anymore. It's dysfunctional because of the dominant negative effect. You can see the red color here is no longer linking to the green color, which is the muscle membrane here. And so normally we get the nice yellow color because the link here and here, it's separate. And so the linkage between muscle fiber and the matrix is no longer there. That has therapeutic consequences. If you have loss of function on both gene copies, so there's no collagen 6 being made, you would think, well, the strategy to take is we're going to replace it with a fully functional gene copy and do gene replacement to restore collagen 6 production. But if it's a dominant negative toxic interference mutation, we need to inactivate the interfering mutant gene copy, the one that messes the normal copies up, because we know just one normal copy is just fine. If we get the mutant copy out of the way, we have a way to therapeutically address these dominant negative mutations as well. So let's look at the loss of function and gene replacement approach here. We have mutations on both alleles. In my little simple diagram, this is shown as an AAV-mediated gene replacement here. AAV is a virus that is inactive because it no longer has a virus genome. Instead, it carries our replacement copy of the gene we want to replace. It infects the cells, delivers a replacement copy to the cell where it is in the nucleus, but doesn't integrate into the chromosomes, makes its own transcript, which causes a normal protein to be made, and then we can ameliorate the disease. The workhorse for this is AAV, adeno-associated virus, a small virus which in itself is not pathogenic. It doesn't cause any disease, but it needs helper viruses to replicate normally. We don't want a replicating virus, so we're taking the helper viruses away, we're taking the virus genes away, and we're replacing everything with a therapeutic copy of the gene that we want to replace. The only problem is that our loading capacity in AAV is limited. It is 4.6 kilobases, and what that means for us I will show you in a second. So you can make that in a bioreactor by putting all the genes that you need to make a virus separately from the gene that you want to deliver. And these genes that make the virus provide the virus capsid, the envelope, and that is packaged with the transgene, the gene you want to deliver. And what you have in the end is viral particles that contain not viral genes, but they contain the transgene that you want to replace. And with the AAV capsid, you can deliver lots of goods for genetic therapies in our toolbox. But we're going to focus now on the gene replacement, which is where you put the missing gene as a specialized copy into the viral capsid to deliver it to cells, and then have the cells make the replacement gene to make up for the mutant one. It's best for loss of function, as I told you, where there is no collagen 6 being made, for instance. Those are the recessive disorders of collagen 6. But in addition, the size has to be right. It has to fit into collagen, into the AAV vector. Once it's packaged and the body is infected with this viral particle that carries our transgene of interest, the viral particle will infect the cells and deliver the gene to the cell nucleus, where it creates its own genetic material. It's called an episome. It's kind of a little tiny extra chromosome where our replacement gene lives and is made into the transcript and the message. Problem is the size restriction. Collagen 6A1 and A2 will fit into this, but not called 6A3. That's too big. And we have to come up with other strategies. Other strategies do exist, so we can think about lipid nanoparticles, for instance, which are not viruses. They're kind of lipid droplets that can carry a much larger cargo. And that would be, theoretically, a way to sneak in even larger replacement genes like called 6A3 and deliver RNA directly that way. That's a principle behind the RNA COVID vaccines, by the way, which are also delivered with lipid nanoparticles as RNAs. So for the dominant negative situation, we have to go another route. We have to inactivate the interferent mutant copy. And this is back to the assembly of collagen 6, and you can see the problem here of a dominant negative mutation. So here are our 3 gene proteins that are made from the 3 different genes. Let's assume on alpha-3 there's a mutation on one of the 2 copies of the alpha-3 gene, shown here. And because we have this assembly process going on from the monomer to the dimer to the tetramer, once this mutation is allowed to assemble, it causes havoc, basically, in the tetramer, where then most of the tetramers have at least 1 mutant chain. And the reason why that is because the dominant mutation that are common in the collagen 6 disorders are all in one particular spot of the triple helical. We call it the triple helical domain. That's a collagen domain. But it's at this end of the triple helical domain where they cause the problem. And the reason why they're allowed to assemble is because the assembly of the collagen 6 genes is like a zipper from the C, from this end of the protein to this end of the protein. And you can imagine when they're zipped together, starting from this, what we call the C domain, zipping up the triple helical domain, by the time they reach where the mutation is, it's too late and the zipper has already closed. The mutation is being assembled and causes havoc in the tetrameric state. And the answer to that would be, well, we have to inactivate the mutant copy. If we take it out and don't allow it to assemble, then we only have 1 copy here of the called 6A3 gene, but it's fully functional. And if that's allowed to assemble normally, then we get less collagen 6, but it's all normal. And that reduced amount of normal collagen 6 is enough to prevent disease. So that's a concept of allele-specific or mutation-specific inactivation of the transcript. So again, here we are trying to go as close as we can to the causation of the mutation, but current technologies are mostly directed at the transcript here, at this part. So this is before assembly into the protein, so still in time to prevent assembly of the mutant protein, and that's transcript-directed approach. Transcript-directed approaches have a big role in the molecular toolbox. They can address conditions that cannot be addressed with gene replacement, such as our dominant negative problem here in collagen 6, and they can be delivered as small molecules, not as viral gene therapy, as small molecules that may be theoretically easier to make and maybe more cost-effective. You can also, because they are more cost-effective, design them as precision therapeutics, even for a single patient mutation. In my little diagram, it would look like this. So we make a little molecule, an ASO or an siRNA. It's a small molecule made of genetic material that goes to the transcript, not to the chromosome, to the transcript, corrects the transcript, and leads to a normal protein. So let's go back to allele-specific or mutation-specific knockdown. The concept here is to interfere with the mutant RNA, but not with the normal RNA from the normal gene copy, to specifically take down the mutation and not allow it to interfere with the normal copy. This can be done on the RNA level with RNA interference or siRNA, but also with antisense oligonucleotides. The concept is always the same: recognize a mutant RNA and take it out. And we've shown that as proof of principle as well. Other groups have shown similar results for different mutations, but I'm just showing this as an example. This is Veronique Boldix and Yatchin Zhu's work, where you can see they created a probe that recognizes a mutant RNA that's missing 1 exon, the exon 16 here. When the exon 16 is present in the normal RNA, it's not recognized by the molecular probe here, the siRNA, and therefore only the mutant is taken down. And I spare you the details here but the upper line here is a normal copy, the lower line here is a mutant copy, and you can see our molecular probe, for instance, this one here, is very good at getting rid of the mutant and leaving the wild type alone. And that actually leads to a nice restoration of a normal collagen 6 matrix. Just pay attention to this one here. So in the mutant case untreated or with a control treatment, you don't see any green matrix here in the culture. But when we treat it with this allele-specific approach, then we restore what looks like a fairly normal looking extracellular matrix by just taking away the toxic product here. When we have that now, as you will learn in Astrid Buhl's talk, we have now been able to refine that to even address point mutations like the glycine mutation shown here with a very high allele specificity. This is shown here in a reporter system, and you'll see more of that in Astrid's talk, where you can see the transcription that happens from the wild type, the normal gene copy, and the mutant gene copy. It's a reporter system here. When there's transcription, when the protein is made, it's green. And you can see this siRNA here turns off the light for the mutant, but leaves it on for the wild type, and same here. And so that is allele specific and quite promising. But again, these are approaches that are really tailor-made for the specific mutation that we need to address, and this is this glycine mutation here. They're really precision medicine down to the single mutation, and therefore only apply to very few patients, sometimes even only one patient, and we need to find a way to do that. Another precision medicine approach that is directed at transcript is splice modulation. And here you interfere with the splicing, or you try to correct the splicing, the putting together of the exons into a sense-making message in the gene. This applies to a number of mutations that cause missplicing, the splicing machinery to go awry. And this is often found with modern technologies in diagnostics such as RNA sequencing. It may be hidden to a regular genetic testing. So really a plea for precision diagnostics. As you will hear from Veronique later, there is one mutation of such splice abnormality that we found recently called 6A1. It causes an abnormal splicing to occur where it shouldn't occur, and it causes a toxic exon, a pseudo-exon, to be spliced into the gene copy from that gene. Causes a toxic version of called 6A1. And what we can do with the splice modulation is we can force that to be spliced out again, to be not included into the protein using small molecular probes called ASOs. And when we do that, we can mask this abnormal splice event. We can coax the splicing machinery into splicing normally and ignoring this abnormal exon and not put it back into the protein. And then we have normal collagen 6 again. That brings me to, of course, the genome editing technologies. You've all heard about CRISPR-Cas-based system, the gene scissor system. This is a very clever system derived from actually bacterial immunology, as bacteria developed this as a defense mechanism against viruses that affect bacteria, bacteriophages. And what it does, it's a search and destroy mechanism, if you will. It has, it's like a search function in a word processor. You have a sequence that you want to find. You search for it with a guide RNA here, and that once it's found, recruits a protein complex, the Cas9 protein complex, to that part of the gene that you want to find, that part of the genome that you want to find, and then Cas9, when its scissor function, cuts in the DNA where it's being directed to. And then what you can do, you can either say, well, the cut is all I need, and that's one possible outcome. But you can also then try to replace it with a normal template and put in a repair patch to where the abnormal DNA was. And that really is the holy grail of this, where you can then not only cut out the mutation, but replace it with normal DNA. This can be delivered to the cells using, again, our AAV delivery system. Now that we don't put a gene replacement in here, we put the molecular tools for the gene scissors, for the CRISPR-Cas system in there with the guide. That's our search function here. And then the executor, the CRISPR system, the Cas9 executor to be guided to the mutation spot in the genome to repair it. This can actually be applied to things like the pseudoexon that I showed you before. This is work from Apurva Sarati in the lab, where she said, oh, we have the pseudoexon sitting here in the intron. We don't want it to be appearing in the messenger RNA because it's toxic. Let's see whether we can use CRISPR-Cas9 to cut it out. And so she used two search functions to search either end of the pseudoexon sequence in between the normal exons, and directed the scissors, the Cas9, to these and cut out the pseudoexon from it. And that was actually quite successful. You can cut out the sequence with the pseudoexon, and I spare you these gels here, just to say that the pseudoexon then disappears and the defect is repaired. So this is just another example of CRISPR-Cas gene editing, where double-strand breaks are introduced by the gene scissors in the chromosomes, and then you put in the repair patch here, where you can replace a missing piece. But in the dominant negative situation, if we successfully cut just on the mutant copy and just inactivate it that way, to really get rid of the mutant copy by gene scissoring specifically on the mutant side of the two genes, then you would have the result that you want, which is inactivation of the dominant allele. If you're able to also repair it, all the better. But in the dominant negative situation, that may not be necessary. And just a precise cut on just the mutation side of it will be enough. And you'll hear about this probably in this meeting as well, including from Cecilia's group. So genome editing is, if you will, extremely powerful. The applications and technologies are really still expanding at a really rapid pace. Every month or so brings a new iteration of this technology. It's getting better and better, and precise and more precise and more precise. But the efficiency and the safety of it are still lagging somewhat behind and need to be established fully to be viable systems for our problems in the collagen 6s. But the good news is, if this tool is developed for other conditions at a rapid clip, then we can use it and just apply it to our conditions just as much. And so any development that happens in this field has a benefit for the collagen 6 disorders in the end. There's a special case of genetic editing, which is called base editing, and I will just mention it briefly at the end here, which is applicable to many point mutations, including some of the mutations that we see in collagen 6, but not all. But what it is, it's basically also a search function, like a word processor search, where you find a bit where you have the point mutation, and then you direct another clever protein that is an editor of bases to this spot, and the editor will edit the mutant base into a normal base. So you really repair that one point mutation with the base editor by bringing it directly to the point where it is needed. So another surprisingly powerful tool. There are variations of that that I will not get into greater detail, called prime editing, where you have the same principle of a search function in the genome, where a machinery is guided to the mutation. Then a cut is made in the DNA, and the DNA is repaired along a template which guides the repair mechanism to put in normal instead of mutant sequence. And so that is very versatile in theory, and is getting more and more efficient and specific. And finally, you can do editing of mutations also on the level of the transcript, so not of the genome, and that has the advantage of being not permanent. So you're not running the risk of making potentially deleterious off-target editing in the genome. This is all reversible, and the safety margin, therefore, is much better. And so here you do deliver your editing tools to the cells, but now the editing is happening on the level of the transcript, not of the chromosomes or the DNA. So you do no permanent damage, but you still repair the mutation before it is made into a protein, and could have an effect like this as well. Finally, it's important to realize, and you hear about this in talks, including from Veronique, it's important to know that we'd have to target the muscle fibroblasts here, the FABs, the interstitial cells, labeled in green here in our work from several years ago, because that's where the collagen 6 is being made. And all the efforts in our day are targeting muscle cells, not fibroblasts. So we need to make a concerted effort to develop variants of AAV, variants of our ASOs, of our siRNAs that are able to specifically also go into these interstitial cells. We don't want them to exclusively go there, but they have to also go there. And this is some promising first results that Veronique generated for siRNAs in collaboration with the Kurova lab in Worcester, Massachusetts. So this is not for you to read, but just to say there is an expanding molecular toolbox of tools to manipulate genetic information, repair mutations, replace mutations, inactivate mutations that we can grab into to apply to the specific problems of the collagen 6 disorders. At the moment, leading is AAV, ASO, and siRNA technologies, which you will be hearing about in this conference. But really the idea is to make this available to a patient all the way to identifying the mutation and bringing it back to the patient as a precision approach. And that does need much more than we can offer at this point in time. It really needs a network of partners that help with production of these agents, regulatory support, new technologies, and clinical sites that can put them back into patients. But I think the time has arrived to make this happening. And I'll leave you with that idea. Thanks.